So in this module, the objective is discussion of isozyme, what is its characteristic, what are the different methods which are used to characterize and separate isozymes, what are the different types of the isozymes, and we will also discuss what is the origin of isozymes. First, we will discuss what are the different types of isozymes. So, isozymes are classified into three types based on their origin and formation. The first type is true isozyme, second is hybrid isozyme, and third is allozyme. Each one will be discussed in detail. First, we will discuss about true isozymes, then hybrid isozymes, then allozymes. What are true isozymes? True isozymes are the one that is a product of different genes and they are generally coded by different genomes. For example, if you remember malate dehydrogenase of the TCA cycle, so malate dehydrogenase is having two isozymic form. One is cytosolic malate dehydrogenase, another is mitochondrial malate dehydrogenase. Both are involved in interconversion of malate into oxaloacetate, but they are coded by different genomes. But if you remember the function of cytosolic malate dehydrogenase and mitochondrial malate dehydrogenase, so mitochondrial malate dehydrogenase converts malate into oxaloacetate, while cytosolic malate dehydrogenase converts oxaloacetate into the malate. So they are isozyme involving in the same step but performing different reactions. Not only that, they are originated from two different genomes. For example, mitochondrial MDH, malate dehydrogenase, is synthesized from mitochondrial genome, while cytosolic malate dehydrogenase are synthesized from the genes present in the nuclear genome. The second example of true isozymes are amylases, which hydrolyzes starch. So amylases is either salivary amylase or pancreatic amylase. So they have difference in the origin. One is synthesizes in the salivary gland and other is synthesizes into pancreatic cells. So true isozyme basically have difference in the genome or genes which are coding that isozyme. The other class of isozymes is hybrid isozyme. As the name suggests, hybrid means they are having different things which hybridize together to form this isozyme. So hybrid isozymes are oligomeric protein that are produced by different combination of constituent subunits. So when we are saying subunit, that means the isozymes which are hybrid isozyme, they are always oligomeric in nature, they are always existing into the quaternary structure of the proteins. So example of hybrid isozyme is well known. You have heard about lactate dehydrogenase. So lactate dehydrogenase is having 5 isozymes. They are formed because of the different arrangement of its two subunit, M subunit and H subunit. So M subunit, if it combined together, then it form M4 isozyme that is found in the muscles. Have only H subunit, it is producing H4 isozyme which is present in the heart. And if we have different composition of H and M, then we will have H3M, H2M and HM3 isozymes. The second example of hybrid isozymes are creatine phosphokinase, which have 3 isozymes. In one isozyme, we have both subunit as a B subunit, while in other isozyme, we have one B subunit, one M subunit, while in the third isozyme, we have only M subunit. So, in this way, you can see that how oligomerization of subunits leads to the formation of different isozymes. Third class of isozyme is allozyme. So, allozymes are the product of different alleles of the same gene. When you are saying alleles, that means alleles are form of the same gene. They are arises because of the minor variation in the genes. So, if the allele having minor variation in the sequences, then your allozyme also have minor differences in the sequences of amino acids, and the differences in the amino acid sequences leads to the difference in the catalytic parameters, and that will have resulted into development of allozyme. One such example is glucose 6 phosphate dehydrogenase. What are the methods that is used to characterize or separate isozymes? When you want to study isozyme, then it is very important to know the methods which characterizes and separates isozyme. As you know that isozyme differ from each other in the structure, it is differing in its electrophoretic mobility, it is also differing its immunological activity. So, therefore, any methods that characterizes structure, electrophoretic movement or immunological recognition of isozyme can be used to separate the isozymes, because they are differing from each other in these properties. As you know, isozymes differ structurally, so the methods that are used to distinguish the structure, they all are used to characterize isozyme, such as circular dichroism. Circular dichroism is a method which differentiate the secondary structure of enzymes or proteins. In the circular dichroism, when a circular light passes to optically active compound, it is converted into elliptical light. So, CD spectroscopy is monitoring the ellipticity of light coming out of the enzyme or optically active compounds, and CD spectra is a plot of wavelength versus ellipticity. So, we have a specific structure spectra for alpha helix, for beta sheet, for random coil. When you compare it with the CD spectra of different isozymes, then you can see the differences in the secondary structure. So, the CD can be used to differentiate the two different isozyme and characterize it. The second method that can be used to distinguish two different isozyme or characterize it, that is NMR, that is nuclear magnetic resonance. So, in the NMR, what we are going to do is, we are basically exciting proton of a nucleus, and after excitation, once the excited proton coming back into the ground state undergoes relaxation. So, the relaxation pattern is monitored in the NMR, and relaxation pattern is different according to the change in the 3-dimensional space around the proton. So, if two isozymes differ in the sequence or in structure, they will have the surrounding of proton different. So, therefore, they have difference in tertiary structure, and NMR is used to differentiate tertiary structure in the solution. Therefore, NMR can characterize two different isozymes. The third method is fluorescence spectroscopy. As you know, the fluorescence spectroscopy monitors the fluorescence of tryptophan of the enzymes, and it is very sensitive to the environment around the tryptophan. So, if there is a variation in the sequence of two different isozymes, then there is a difference, there is a variation in the fluorescence of the tryptophan. So, that will used as a probe to monitor the differences in the tertiary structure of isozymes. So, therefore, fluorescence can be used to distinguish two different isozymes. Similarly, we have another spectroscopy that is known as infrared spectroscopy that also monitors the secondary structure of isozymes or proteins or enzymes. So, therefore, infrared spectroscopy can also be used to distinguish, to characterize two different isozymes. So, all these methods which are used to characterize isozyme based on the structural differences in two different isozymes. After structural characterization, we can characterize isozymes based on electrophoretic mobility. So, the electrophoresis separates molecule on the basis of charges, shape and size of the molecule, and isozymes differ from each other in amino acid composition. Therefore, they will have differences in the charges as well as their sizes. So, they can be easily distinguished by electrophoretic movement. As you can see in this figure, where there are 5 isozymes of lactate dehydrogenase, they are shown here, the H4 form, M4 form and other isoform. You can see that they have difference in the electrophoretic mobility. Not only that, the expression of a particular isozyme of lactate dehydrogenase can be seen as the density or thickness of the band. If band is thick, it shows that particular isozyme is expressing in that cell or cell tissues. For example, if you see the liver matrix, if you see the heart cytosol, you will find that in liver we have M4 form that is expressing, while in the heart cytosol it is the H4 form that is dominating. Even in the liver cytosol we will have M4 form that is dominating. Isozymes can be easily purified by using affinity ligands. So, as you know that affinity purification is the best method for separation of two different molecules, and isozymes differ in the sequences. Therefore, if we will generate that sequential antibody can be used as a ligand in the affinity purification, and that ligand is helping you to separate, to purify isozyme. So, you can characterize isozyme using the structure based method like CD, fluorescence, infrared spectroscopy and NMR. You can partially purify isozyme by using electrophoresis method, or you can purify it by using affinity purification, which is based on sequential antibody designed against isozymes. So, after discussion of types of isozyme, methods of separation, we will discuss now the significance of the isozymes. So, different isozymes have many significances, and their significances are grouped into physiological significances, embryological significances, clinical significances and metabolic significances. We will discuss it one by one. So, the first significance, that is physiological significance. So, isozymes are adopted for the survival at different physiological conditions. For example, lactate dehydrogenase have different isozymes and that are needed for survival in different tissues with difference in the physiologies. Lactate dehydrogenase catalyzes the interconversion of pyruvate into lactic acid by using NADH. So, pyruvate is converted into lactate and utilizing NADH, which is converted into NAD. Different isozymes of lactate dehydrogenase can be synthesized in the body based on the oligomerization of two subunits. The first one is M subunit, that is known as muscle subunit, and the second is H subunit or heart subunit. The 5 hybrid isozyme LDL, LDH1, H4 heart, LDH2, LDH3, LDH4 and LDH5. So, these form are designed because of hybridization of different subunits. LDH1 found in the heart and LDH5 found in the skeletal muscles, while other isoform also have distribution in different tissues or different physiologies to perform a specific physiological requirement of that tissue. The second significance of isozymes are clinical significance. So, the presence of different isozymes have clinical significance. For example, creatinine phosphokinase, which is involved in the conversion of creatinine and ATP into the creatine phosphate and ADP. And creatine phosphokinase is a dimeric form, consists of a brain subunit and a muscle subunit. So, 3 types of creatine phosphokinase having different arrangements of brain subunit and muscle subunit. So, CPK1, which is having 2 brain subunit, CPK2, which have 1 brain and 1 muscle subunit, and it is found in the heart, and CPK3, which have both muscle subunit. So, as you see here, that CPK2 is present in the heart. So, if normal level of CPK2 increases in the blood, it shows that there is some problem in the heart, and normal level of this isoform is 5% to total plasma CPK, and that is increased to up to 20% under myocardial infarction. So, other isozymes are also used to monitor some pathological conditions. They are either isozymes, either not isozymes, but they have significance in monitoring the pathological conditions such as SGPT, SGOT, LDH, ALP and GHT. Embryological significances of isozyme. Different isoform of biomolecules having embryological significances. One such example, which is not an enzyme but that is isoform, is hemoglobin, which exists in different forms in embryological state and adult state. In embryological state, we have alpha 2 gamma 2, while in the adult state we have alpha 2 beta 2, and both have its own significances. The fetal hemoglobin or embryonic hemoglobin have more affinity for the oxygen, hence help in the survival of the fetus, but it has less oxygen delivering capacity, therefore that cannot be used in the adult. Infants or fetal physiology is in such a way that fetal hemoglobin can deliver oxygen to fetus, but they cannot deliver oxygen to the adult. Therefore, in the adult, that alpha 2 gamma 2 is replaced by alpha 2 beta 2, which is having more oxygen donating capacities. Isozyme also have metabolic significances. For example, the existence of isozyme permits the tuning of metabolism to meet the particular needs of a given tissue, such as hexokinase and glucokinase are 2 isoforms. Glucokinase found in the liver and pancreatic cell, and it have high Km for glucose, that means it has a less affinity for glucose, while hexokinase, which is found in different tissues, have low Km for the glucose. Therefore, the existence of hexokinase in all the cell promote glycolysis in that cell, while existence of glucokinase with lower affinity of the glucose promotes insulin release by beta cell of pancreas or initiation of glycogen synthesis by the liver cells. Both these processes must occur when glucose in abundance. Therefore, glucokinase is not present in those cells, then synthesis of glycogen and release of pancreatic insulin doesn't take place. Therefore, the existence of isozyme basically helps the metabolic pathways and fine tuning of metabolism. Isozymes traditionally also used as a molecular markers or biochemical markers, but nowadays with the development of different methods, molecular techniques, they are of less use, but traditionally they are used as a molecular marker. So isozymes have different significances such as physiological, embryological, developmental, and then clinical significances, and they can also behave like molecular markers. After discussion of type, significances, and methods of separation, the important point is how the different isozyme originated. So isozyme usually result from the gene duplication, but it can also arise from polyploidy. During the evolution, if the function of new variant remain identical to its parental variant, then it is likely that one of the two variant will be lost and resulted into the pseudogene. But if mutation or variation do not prevent enzyme from functioning, then instead of modifying either of its function, then two variant may both be favored by natural selection and become specialized to different functions, and that is because of amino acid residue change. So if the amino acid is changed, which is relatively unimportant, then it is selectively neutral, then it is subject to genetic drift, but if and undergoes natural selection. So that means you can say that because of the mutation we are creating the variants, but if variant is performing some other function, then that has been selected from natural selection, but if they are not performing any other function, they are considered and they are produced, they are converted into the pseudogenes. So allozyme may result from point mutation, from insertion deletion mutations after DNA coding sequences of the gene undergoes mutations. So students, let's summarize what we have discussed in this module. So in this module, we have discussed what is really a isozyme, what is a true isozyme, what is hybrid isozyme, what is allozymes. We have discussed how the trio isozyme differ from hybrid isozyme and how hybrid isozyme differ from allozyme. We have discussed that trio isozyme have different origin, hybrid isozyme have different oligomerization, and allozyme originated from different alleles. We have also discussed the clinical significance, physiological significance, embryological significance, and metabolic significances of isozyme. We have also discussed in this module what are the methods that are used for characterization and separation of the isozymes. Not only that, we have also discussed about the different monomeric composition of lactate dehydrogenase, which is a hybrid isozyme, creatine phosphokinase, which is also a hybrid isozyme, and what are their clinical significances. We have also explained what is the origin of the different isozymes. So, from this module, you have learned the types of isozyme, significance of the isozyme, origin of the isozyme, and their separation and purification methods of the isozyme. This will help you to understand the clinical significance of the isozyme and their development as a marker to monitor the different pathological conditions. Thank you.